Hi everybody and welcome to the Kidney Coach YouTube channel. I am naturopath Fiona Chin, co-founder of Kidney Coach and Kaigenesis, a range of supplements designed to support those diagnosed with chronic kidney disease. And I'm joined again today by the wonderful, the amazing, the fabulous Felix Mueller. Felix is coming in all the way from Germany. I don't often speak to the amazing Germans, but I have a very high regard for them because they're highly intelligent creatures. Felix, if you didn't watch a previous podcast, I would call him the absolute expert when it comes to things like polycystic kidney disease. And I'll say that because even though Felix might not have this huge background in trained science, he's a lived experience of PKD. He has a bit of a brain-like mind. He loves to go down rabbit holes and understands that everything should be curable and will just turn over every stone until he comes up with things that could help the community and himself. He actually managed, and he's one of the first people I know, and we spoke about this, Felix, last time we did a podcast, that you'd actually managed to reduce the size of your PKD cysts that was shown on a ultrasound, which is kind of unheard of in PKD because it's meant to be this progressive disease. They're meant to keep getting bigger. Felix did a whole pile of research when it comes to using ketogenic diets and how to put them in for people to actually reverse PKD. Then he's created this amazing website, revertingpkd.com. I'll put all the links below. But he's, again, because he's got this amazing brain and he just never stops trying to uncover stuff, he's made some other pretty cool connections, which is, he reached out to me, he's like, we've got to talk about this. And he just filled me in before and I'm like, yes, we do. So Felix, I'm going to turn it over to you to talk all things gut-kidney axis and the relationship between the gut, the microbiome, cancer and PKD. Let's tell everybody all this amazing stuff that you found out.
Yeah, thanks for having me. I'm very happy to be back. I enjoyed our last conversation. We went pretty in depth there on all things ketogenic diet and PKD. And that was sort of my start into this whole sphere. Me having PKD, having discovered the ketogenic diet, sort of building a community. I also have this Facebook group called Healing Polycystic Kidney Disease Naturally. And I sort of started publishing all the stuff on what I found with ketogenic diets over there. And we've since, got a lot of other people who have reduced kidney size, who have improved their kidney function. So yeah, it's a system now, which is great. It's all based around the Bulletproof Diet, which is a great book written by Dave Asprey, who also wrote the foreword for my book now. And it's a very detailed way to look at the ketogenic diet, which removes some of the toxic parts of it, and is a lot more specific on like how to remove plant toxins, how to remove human-made toxins like pesticides. So all of that sort of gives us a little bit of an insight into, okay, we can do something about PKD. And the mechanism that I started with was very similar than what we see to what we see in cancer. So Professor Thomas Seyfried has published this book and a lot of studies on the metabolic workings of cancer, right? And he found out that the cell, basically it has two modes of making energy. So it can either make energy respiring, so producing energy with oxygen and glucose, and this process is very efficient. So it makes about 32 to 36 units of energy, which is ATP, is the name for it. So this is very, very efficient. And then there's another mode, which just uses the glucose, and it's very lossy. So it only produces 2 units of energy, and it produces a lot of byproduct, which is lactate. You can feel when that energy production mode is active, when you're sort of overexerting yourself in the gym, for example, and you sort of feel that burn in the legs or in the arms. That is lactate, right? And that is only there because this mode of energy production is fast, but it's very lossy. So Thomas Seyfried found out that in cancer, the cells have access to oxygen, but they're not using it to produce energy. Instead, they're sort of not using it, and they're just using the glucose, which leads to this lossy type of energy production. And the problem is, in this mode of energy production, the body links growth and energy production. So that means we have to because we're just making 2 units of energy instead of 36, we have to upregulate this process by 18-fold. So that means we're upregulating growth by 18-fold. And this is why you get a tumor. So, of course, we're talking about PKD here and not cancer. So why is this important? Well, in PKD cysts, we're also seeing this uncontrolled growth. It's not always linear, it's not always the same, but there are periods of time where these cysts are growing, and in those periods of time, the cysts are using this same mode of energy production. This has been found by testing the cells. You can stain them and see how much glucose they're uptaking, and we can see that they're taking up a lot of glucose compared to healthy cells. So we know that they're in this mode of weird energy production, and we're asking, why is this happening? So there was another hint that I found a long time ago, and I couldn't really, put it into the model yet. I didn't really understand how it all fit together. So in the 80s, there was an amazing study on a PKD rat model or mice model, and they had these mice that were germ-free. So they didn't have any gut bacteria or any bacteria in their body for anywhere. Like now we know there's even a kidney biome, right? There's bacteria everywhere. But these mice didn't have any bacteria. So 96% of them had no cysts, even though they had the gene mutation that mirrors PKD in humans.
Okay.
Which is already amazing, because in theory you could try to produce a germ-free human that has PKD and sort of stop it. But of course nobody's done that, and then you have to ask the question, what is it? What part of the germ-free process makes this PKD thing stop? And we didn't know back then, but then there were a couple of other studies done in the petri dish, where they tried to generate PKD cysts from mutated tissue. And you always have to have some kind of initiating event, right? If you just grow cells from PKD people in a dish, they're not going to have cysts. They look normal for a long time. So they want to speed up that process, right? So what they're doing is they're injuring the cells. They're putting some chemicals like NDGA is one that they use. And then they try to create injury with that on the kidney cells, and they want to provoke a cyst growth. But they found out that it's not happening. They can't get it to grow the cyst. So what did they have to add for the cyst to grow with injury? It's endotoxin. So endotoxin is a toxin that is made by gram-negative bacteria, and it's also the outer shell of the gram-negative bacteria. So it's sort of the same thing. It's sort of shedding part of its skin. If it dies, it sheds a lot. As long as it lives, just sheds a little. And this endotoxin was required for the cysts to grow in the petri dish. So then you ask the next question: do PKD patients have more endotoxin?
Good question.
Yes. If you look at PKD patient urine, most of them have elevated levels of endotoxin. Now, that's pretty interesting. And, well, the question is there maybe some type of element in the gut flora that is predictive of PKD cyst growth? And I have actually put out a lot of this in my book already. And a short while after that, there was actually one study, and I think it was also out of the Wimes lab, that looked at part of the gut flora. And they actually did find that a couple of species were routinely missing in PKD patients. And I think it might be worthwhile to load up on that species. I forget the name right now, but it's out there, and I can find it after this. We can link it below. Which is an interesting aspect. So, but we don't know, right? And then you look at population sequencing studies. So there's studies that look at, like, in 10,000 people, how many people do have the PKD mutations. And you would expect that it's about 10 people in 10,000 from the ultrasound sort of statistical data that we have. But if you do that study, and it has been done, it's been York Pye has done it with his team, then you find it's actually twice that. It's 20 people in 10,000. So
Okay.
we don't know. And I've tried contacting these people. I've gotten no replies. I don't know why. It's very frustrating. But I think, and I would love to, confirm this or test this theory, that the reason that there is twice as many people in the sequencing studies as in the ultrasound is that there's people who have the gene who don't have cysts.
Yeah, and if you wouldn't know unless you had a family member or you were tested, and we know, especially in America, a lot of people with parents with PKD don't tell their children because of the insurance issue, and they just hope that it never happens in their children. I've spoken to parents of that, and they don't want to scare their children, and they won't get insurance if they've got PKD. So a lot of people aren't testing. So it's interesting, you're right, just because you have a family member doesn't mean you'll often get it, and often people aren't screening for the genetics. So it's fascinating you say that. So then, Felix, what's the next question after that? If we've got half the population with PKD, but double the population with the genes, maybe, hypothesis. There's a mechanism, obviously, between these endotoxins that almost is required, tell me if I'm hearing this wrong, is required to switch on the process of cyst growth. And without that switch, potentially PKD or cysts won't grow. Hence, meaning if you could find the off switch, we might have a potential treatment. Have I concluded correctly?
Yes, you have concluded very, very well. And I think we actually know how that switch works, and I'll explain it.
Yes, please.
I actually have one client who has a brother who's older than him, who has just one cyst. And they always assumed the brother didn't have the gene. I think he might have the gene. And I think he might just have the perfect gut flora to not trigger cyst growth.
And it's interesting, you said he was the older brother, right?
Yeah.
Which, when you know about, depending how they were born and the mother and the microbiome and how it gets passed on, he's in a better position to have a better microbiome generally, if all things presented equal, being first child. So that's fascinating.
Yeah, and like, to give the conclusion away in the middle of the interview or whatever, I think, like, if this theory is correct and we can sort of validate it, then we might just be able to, with population sequencing, or even, with this guy's brother, find the gut biome that doesn't trigger PKD, which would then mean we could do fecal transplants to fix cyst growth. Yes, exactly. And I'm gonna tell you why I think that would work.
Please do.
The big question is there a gut microbiome that is so non-toxic, that it doesn't produce this stuff, that might trigger it? And I don't know if it's just endotoxin, but it's definitely also endotoxin.
Yeah.
So, in PKD, the proteins that are mutated, they call them polycystins, right? Polycystin 1, polycystin 2. It's such a weird name, because where else do we name the protein, or like the thing that should do something, after what happens if it doesn't work?
Yeah, that's actually a really good point.
It's such a weird. So I came up with a new name.
I love it.
It's called the Clear 1 and the Clear 2 proteins. It's an acronym for calcium level and epithelial adhesion regulator, because that is what they do.
This is a much better name.
Yes.
Let's put it in science books.
Yeah, I think we should rename them, actually, because once you hear that name, then you start thinking about the right questions. Okay, so these proteins, they sit on the outside of the cell, not just in the kidneys, in every organ, and they regulate the level of calcium that gets into the cell, which is very important for energy production, which sort of, if you have too little of those proteins, which is the case in PKD, actually they're probably not misfolded, not misformed, it's just less. Okay. So, I spoke to a researcher friend of mine recently, and he explained to me, which I was not aware, that if you're heterogeneous mutated, which means you just have one copy of the sort of normal version and then you have one mutated one, that you're probably not expressing the mutated version.
Yes.
So you probably just have about 50% of the normal amount. So it's not that you have wrong formed. So I had a misunderstanding about that earlier. So at least that's what he's saying, and I'm going to take his word for it. I wasn't able to find out a lot more of that yet. Anyway, so it sits on the outside of the cell, and it's sort of like this the clear 1 and the clear 2, so PC1 PC2 proteins, and they together, they're sort of one is sort of saying, okay, now open the gate, now close the gate, and the other one's the gate. Okay. So that way we already have like less calcium inside the cell, which means we're prone to less energy inside those cells, because calcium is needed as a catalyst for that whole energy production system that we talked about earlier. But then also it's epithelial adhesion regulator. So this means that we have, like, this is very known in the gut area, which is relevant. There's something called leaky gut. Mm-hmm. You have those cells, should be tight junctions is what they call it. And if you have leaky gut, they're like a little bit more farther apart. And the stuff leaking through, and you get allergies. That's how you get those. Mm-hmm. So not a great thing. And if you have less of the clear 1 and clear 2 of PC1 PC2 proteins, then you have leaky tight junctions, not in the gut, also in the gut, but not just there, but also in the organ lining and the lining of all the epithelial layers. So for example, the layers that form the outside or the inside of tubules, right? Mm-hmm. So this means that if you have some kind of toxin that wants to be filtered by the kidney, and you have too little of the clear 1, clear 2 proteins, it sort of goes in between the cells where it shouldn't be. This is similar to when you pour stuff in your drain, but your drain's leaking at the bottom, and you sort of have it dripping into your cupboard underneath there. So now imagine the cupboard doesn't have a door that you could open, but it's just closed, and there's nothing where you could put it. So what would you do? You would take a little garbage bag, put the stuff in there, close it up, and sort of fix the contamination problem that way. And it looks like that is exactly what the kidney is doing. So
hence it's putting us back in a cyst.
Yes. PKD kidneys have leaky tubules where toxins go through, and once it's inside this interstitiary area, we have to contain it in a little sac. And the body does this in other places. It's very well known, right? So if you got cystic acne, for example, you get the cyst because there's an infection. Mm-hmm. And the body's encapsulating it, and best case you can sort of pop it open and then you get to the outside. But in the kidney that doesn't work. So we have these forever sacs there. That has also been tested, also in the 96 or something. It's been a long time. They've taken the cyst out of a human, they put it in a petri dish, and they took a syringe and they removed the cyst fluid. They put salt water back in. Rinse it a couple times, put salt water back in. So they displaced the toxins, removed the toxins. 72 hours later, the cyst became normal working kidney tissue again. I've
seen that study. It's crazy. Yeah. It's very cool.
Isn't it isn't it amazing?
It's so amazing. And I mean, I love how your brain works because you're very pragmatic and logical, which means that you see things that people in it often don't see, and you can work your way through, which I would say is a very good series of questions of why, working backwards. This seems logical, and at least that's how I see it. My brain's a little bit similar, maybe not to the same degree, but it's interesting. Now, tell me, and maybe I'm jumping here, the link to cancer, because cancer, this is where people will have done all the studies on, because, there's more money, for want of a better word, there's more money to be made in cancer versus PKD, but it's a very similar mechanism, right? So walk me through that bit, unless I've jumped too far in front.
No, that's it's a good link there. And actually, cancer, there's a lot of money in cancer, but also there's a lot of politicization in cancer.
Yeah, correct.
So I've talked to someone who has written, I think, the most comprehensive and best book and most recent book on cancer that's out there. And I've actually told him, you might be able to do some surrogate studies in PKD, which is not politicized. So that could also be an interesting crossover. But of course, we're jumping the gun here. We got to explain the connection first. So in PKD, we have this energy production problem, which is now we know it's triggered by actually it's not an error. It's a necessary biological function. I think if you were able to block cyst growth chemically, I think you would actually poison the person getting the treatment.
I'm glad you said that, because my brain had gone there too. I was like, if this is a natural mechanism of the body, what happens if we do block it? Where does that all go? What's going to happen to the endothelial tissue? You're going to have decaying kidneys in a sense. I don't know, but
yeah, worst case would be sepsis.
Yeah,
you could die. I would never test that drug. I would never take it in any kind of even end stage. Maybe if you're on dialysis, you could test it, but it's very high risk. And I think we have, as humanity, we have to come to the point where we understand that the body doesn't make any errors. We just have to understand why it's doing what it's doing, and then we have to find the source of the problem, and not sort of tape the fixed engine light.
Totally agree. Yeah, exactly. Cover that up. We won't look at that bit.
Yeah. And we thought for a long time that chemically blocking cyst growth would be sort of addressing the root cause, but it's not. I think it's not. So back to so Professor Seyfried is the one who discovered this whole energy metabolism thing in cancer, which I then found somebody in the literature had also, in 2015, had applied to PKD, which then sort of led me to the idea, okay, we could try ketogenic diet, because that usually fixes that in cancer, so why shouldn't it work in PKD? So in this book where he talks about this whole energy metabolism thing, he doesn't address how it starts. Similar to PKD, nobody talks about this, leakage issue. So there was a book recently released by Mark Linterns called Cancer Resolution, highly recommended read. And interestingly, Mark Lintern, also graphic design guy. I'm a filmmaker by trade. Yeah. So also very different type of mind than scientists. So very broad, not as, deep in one topic, but then sort of connecting. And he actually came up with a new cancer theory. He actually was able to get it confirmed by a big panel of oncologists who voted that it was just as relevant as any other cancer theory. I think it's the most relevant, but I think that's probably the politically correct way that they had to phrase it. And he went even deeper with this. So when I read this, I was like, oh my God, this is exactly what I'm saying is happening in PKD. And now I know probably why it's happening. So he went even one level deeper. So because we're still, we know the body's intelligent and it's sort of trying to stow away the toxins, but we don't know molecularly what's happening there. But he found out that something very similar is happening in cancer. And actually, he might now be of the opinion that a cyst is sort of an immature cancer, so that cancers might start as microscopic cysts, and then when the body's not able to resolve it, then it becomes a tumor. But that's that's just a thought, but not my thought, his thought. So he looked at a lot of research to find out what is the genesis of the tumor. Why does it start? What is the inciting event? Okay, we know that sort of the immune system is not doing everything that it should in that area, but why is that? What starts it? And he actually found out that there's something that's called the cell danger response. And it was not invented by him, or invented like nature invented it, but like discovered. The guy who discovered it's called Navio, and he discovered that whenever a cell has to defend itself against an invader, and the immune system has failed to remove that invader, it's already in the cell, immune system has not done its job. Then the cell has one mechanism left. That's the cell danger response. And it works by taking the oxygen that usually would be used for energy production, making a free radical from it, and blasting that at the intruder. So there's no oxygen left to make energy, as long as it's not fixing that problem. And we can assume that 99% of the time the cell danger response is actually successful, because we've all heard this trope that everybody has cancer 10 times a day or whatever, have a little tumor, and then it goes away because the cells sort of deal with it. And that's how they deal with it. So for a short time, they go to this energy production, the glycolysis with uncontrolled growth, because more cells can do more of that free radical production. So it's basically building up an army, which is the tumor, to attack whatever is encapsulated inside. Very similar to what the cyst is doing.
Yeah, right.
So the cell is not like it's lost its way in energy production. It's like it's using the oxygen for something else. It's using it to attack the invader. And in the case of cancer, he found out at least according to his theory right now, in 100% of cases, it's a fungus that is triggering this. You get a tumor.
That's a big percentage.
Yes. And he's saying, whenever you're for example, you can get cancer if you have radiation, right? That kills the immune system in that area. And then the fungus that's already there, it's a commensal fungus. We all carry it. Candida, I think, is the one that he has in his crosshairs. Candida is always somewhere around. And as soon as the immune system sort of gives way and the cells become weak, then it can start to grow and start to invade cells. And actually it sort of weakens that lining. And then it goes in there and it has a very easy time to sort of hijack these cells. He calls it hijacking. Maybe it's a voluntary response by the cell because it's trying to defend itself. But in any way, it is trapped in that loop of trying to fight off the invader with free radicals, with shooting oxygen at it, and it's not working fast enough to actually kill the invader. So in the case of cancer, we actually we went to a cancer event in the UK that we attended in the audience. He wasn't yet in the panel. I hope he's going to be there this year. And there were two stories that the attendees actually were telling, which were so important. And so now we're going a little bit towards cancer, but I hope that's okay for 5 minutes.
Yeah.
Because nowadays everybody knows someone that's affected. So
yeah.
And people in the audience were very aware of ketogenic diet for cancer. A lot of people had done it. A lot of people in the audience had cancer currently. And so one story was, there was this woman, she was a friend of the person attending, and she had been treating her brain cancer with ketogenic diet very successfully. She she was in very extreme ketosis. So she had like levels of 4, 5
Oh, yeah,
millimole.
yeah.
So very extreme, like this is starvation level ketosis, right? For years.
Oh, wow.
And she managed to like really get rid of the so nothing left on the scan. You know, it was like a tennis ball and then it's zero, right? Not showing up. So great, right? You would think that's amazing. Recurrence probably not going to happen, right? Then she went from a 5 millimole ketosis to a 1, right? She just loosened up the diet a little bit. So what we're doing in PKD,
Yeah.
right? Just like run-of-the-mill ketosis, not like super horribly starving, but still very low carb. Still have to have very little carbs to get there, like 20 grams, 30 grams. And she had a recurrence, and she was dead in 3 months. So imagine like, over years, from tennis ball to zero, and then in 3 months, enough to die from it, even though she was still in ketosis.
Wowzers. That was a resilient little sucker, wasn't it?
Yes, but also it confirms what he's saying, which is if we're using ketosis to block the ability of the body to fight the freaking fungus, you're going to have a lot of fungus,
I see exactly what you're saying, which is the same thing about getting rid of this. Yeah, right. Okay. So the fungus almost in the sense just kept doing its thing in the background.
Yes, it explodes like unmitigated.
Yeah, and as soon as you get like you move that pathway away, it's just like it's tripled in its size. It just hasn't manifested the growth because that's not what they've been able to do. But the actual fungus is okay. I missed that bit. So the fungus can grow still in a ketogenic diet. It just isn't producing cancer cells, and so the immune system isn't getting rid of it. Okay, yeah, I missed that part of it. Yeah, right. Okay,
We all did. We all missed it.
yeah. So right, which is so huge now in cancer because now we're like,
Yeah,
So then my thing is, are we doing people a favor putting them into ketogenic diets?
exactly. I mean, the body obviously can survive a long time just with the fungus. You know, it doesn't seem to kill people. It just probably makes you very yeasty and tired and but you can probably live with that 20 years. So we might be doing them a favor, but we could be doing them a lot more of a favor if they get sort of and I'm planning on getting a very, very, very important podcast going with the guy that used to treat me, who's a holistic practitioner in the UK, and Mark. I want to have them both on because this guy is very good in treating mold, treating fungus, right? So I want to get them both on and have them talk to each other, and I'm going to be like the moderator, and I want to have them sort of figure out like how we treat this thing.
Yeah.
So, but for now, methods exist to get tested for fungus, right? You can test it in urine. My favorite one for that is the Mycotox panel by Microbiome Labs. There's a great book on how to actually apply the treatments, which is called Toxic. Neil Nathan, you probably know that one. I got the tip from Jess, actually.
Yeah.
Revelation, that book for me. Actually, also, it's interesting. I also have aspergillus. So who knows if it actually is also a mold connection, a fungus connection in PKD. I haven't excluded that yet. I don't know that yet.
A lot of my PKD guys will test positive for mycotoxins. It's a panel we run. But yeah, I couldn't say 100% of the time, but definitely mycotoxins. And I can tell you so many of my patients, I screen all of my patients for Candida, and people can be asymptomatic and we'll screen for that.
Yeah. I think
it's the bulk of what I spend myself doing before I do any other treatment. We get rid of Candida until that's clear. The next thing, that's just a standard naturopathic 101 thing. So that's fascinating, not with this in mind, but just because the immune system isn't freed up to do anything else until you get rid of that.
Yeah. Do you also test your patients for the like anything genetic?
Yeah, so I run a full genetic test on all of them.
Do you check for HLA-DR?
Yes. Schumacher
So
protocol, D2, and then the whole pathway to see whether they're, because I used to do a lot of Lyme disease, so you can tell whether people can clear, like the Schumacher stuff, whether they're clearing mycotoxins or Lyme.
yeah, yeah. So you probably see a higher mycotoxin load with non-HLA-DR possessing.
Yeah, and definitely those Lyme chronic fatigue patients, that will come up in, that's the population I've been looking at it in versus like cancer and other bits and pieces. But yeah, those guys generally all have one or two or a combination of.
I think that's so relevant. I think we might find that there's a lot of connection between that. Even, I mean, I would imagine someone who doesn't have HLA-DR to have a worse progression, even in PKD.
Yeah, makes sense now that what you're saying. So we spoke before we hit
Check that in your client
I
database.
could. I actually probably could run it through. Now we've got AI, I could probably pull all of that, take everyone's things off and actually do an analysis of that and go right through it, because it's,
Interesting.
especially in my older cases, every single person was tested on it, and all my guys now go through a full genetic testing as part of just standard intake. So interesting. So tie me back to the gut and the microbiome, because you made a really interesting point, I think, before we hit record about FMT as being a potential, for people that don't know what that is, micro transplants. So loop me back to this kidney gut access, because Candida, obviously, women think about it living in, vaginal tissue and yeasty stuff, but actually it loves residence in the gut. So, and it can often be very asymptomatic. People might just get a little bit of bloating with gas, but people put most of that down to normal, and you can be asymptomatic with Candida. Like I say, I would say 80% of my patients will test for, I test food allergies, and so we're testing IgG, IgE, IgA reactions. 50 to 70% will have an immune reaction to Candida.
Yeah.
So the immune system has picked that bad boy up and flagged it. So that's hence why that's the first thing I do, is try to bring Candida antibodies down. So talk me through this connection in the gut and how you think potentially there's a potential treatment or something with PKD that people actually should start to look at.
Yeah. So and before I do that, I want to mention a second story from the
please.
Just very briefly.
No, please.
The person was trying to treat it with keto for, I don't know, a year, didn't work, got a fungus treatment for something else, and then it worked.
And I'm going to do a side story here too. I treat a lot of MS patients. Now, different mechanism, but you're talking, of course, I go straight to going, how do I apply this to my autoimmune MS guys? I know she has a fungus because she has the weird fungus stuff. We test for it, it comes up. It's been completely resistant to treatment. We've done Schumacher, we've done everything, we've thrown the whole thing of it. We just did low dose immunotherapy using that, and we finally found a doctor that put her on a different type of antifungal. And for the first time in years, and she's so compliant and everything, like we took out microtoxins everywhere, her MS symptoms have resolved.
Oh, really? Wow.
It's using an antifungal that we hadn't used before. We'd used other antifungals. We'd used the Clostridium binders that you normally use in the Schumacher protocol, but it wasn't until we put in a oral antifungal that and she's very MSy, like brain fog, fatigue, dizziness, and like I say, super compliant patient. She's my most compliant patient. Yeah. All her MS symptoms have disappeared.
That's amazing.
So
I wasn't aware of that connection.
Well, yeah, it's one that I'm trying to work out the mechanism as you talk, but I'm just like, I think this connection might go beyond, with that immune system misfiring, it might be beyond just the scope of PKD and cancer. But definitely, I would say there's a huge percentage of my MS patients where they come in and I'm like, this is fungal, this is mold. They'll often have this weird skin stuff going on that, and yeah, you're battling after Candida, and we often have to put in hardcore drugs, antifungals, and my herbs just won't touch the side of it, and then they start to resolve. So fascinating.
Wow.
And the cell danger response, because we see the cell danger response in MS. Macrophages don't switch from M1 to M2. They stay stuck in that mode, which I think triggers this autoimmunity. So how that relates to myelin, no idea. I haven't gone there yet, but it's just fascinating. I do see this carryover in autoimmunity.
Is M2 your word for respiration?
There's a macrophage 1 and macrophage 2. So my understanding of cell danger response is macrophages are meant to switch from an M1 to an M2, which is where the macrophages switch off the cell danger response and they get stuck in first this M1.
Oh yeah, okay.
Yeah, so
they're gonna
yeah,
resolve.
yeah,
They should be resolving, but they're not.
Yeah, they just stay in turned on mode.
Yeah.
Which is that respiration process that you're talking about. So yeah, it's fascinating because that is definitely a pathway in potentially in autoimmune diseases. Is a theory that cell danger response is part of that mechanism. So, but again, no one's gone down the deep rabbit hole. So it's fascinating.
Yeah, no, so I think fungus testing. I mean, there's so many different ways of it, but like if I had cancer, then I would probably do all of them.
Yeah.
Do the urine, do the saliva test, do the blood testing, do the stool test, to do all the fungus testing and, get it resolved. And there's practitioners who can do it. It's not like we have to reinvent the wheel for cancer. Do that. Also do keto because it's also antifungal, which is probably why it's anti-cancer. But it's also sort of, it's just part of it. It's also pressing down on the brakes of the actual metabolism. Yeah, fascinating. So we want to talk about the fecal matter.
And the gut. Yep. Let's loop it back around to this kidney gut.
Which interestingly is the name of a new, or maybe it's not new, but it's a French fashion brand now.
What's the brand called?
Fecal matter.
Matter, right? Yeah. No.
Yeah, and actually, our friend, the biohacker Brian Johnson, he actually was the model for it.
There you go.
Yes, I have no words. So anyway, so we are seeing people who in the statistics who have the gene mutation that is known for PKD, but it's statistically a lot more of them than we would expect from ultrasound population studies. So this might indicate that there are people out there who are like 50 or older and don't have cysts, but they still have PKD mutation. Or maybe they have one cyst, like the brother of this client of mine who, I've talked about. And I actually want to sort of get him to test his genes because he hasn't done that yet. So maybe that would be even simpler than getting the population data. Because I think that's anonymized and you can't really use that to find the actual 20 people who positively tested. But I think if we were able to find this unicorn, which might not be that rare after all, but maybe it is, that has a PKD mutation but doesn't have any cysts and is in progressed age, so like 50 or older, then I think the most likely reason for that being the case is that he has a very good gut microbiome. And actually, my client has confirmed that his brother was breastfed and was not a C-section.
Right, they were the questions I was alluding to earlier on, which are the things that and if you've got the FUT2 genes, the FUT secretor genes, they make a big difference too. I'm sure you're testing those. That bifidobacterium and the bifidolongums, like all the blue zone longevity guys, have a huge amount of bifidolongum. It seems to be a big key species, and if you've got FUT2 genes, which you get COVID vaccines and things like that, will destroy your bifidobacteria and things like that.
Oh yeah, really?
Yeah, so super fascinating that that's what I was alluding to when I asked, being the elder brother, they normally aren't C-section, natural birth, they get the vaginal microbiome population through all of that sort of stuff. So yeah, they're generally at a advantage. Yeah.
And I was not a C-section, I was breastfed, but still, cysts, but it can be destroyed at any point in life. It can be destroyed by antibiotics very early, right? You can get them in childhood, and then you never know if you would have been one of those. Sorry, you never know. You would have been one of those people who wouldn't have grown cysts otherwise. So yeah, I think if we find those unicorns, then we might be able to replicate that by getting fecal matter transplants from those people. So maybe, and that can then be studied, and if we study what's different about those, then we can probably find out what is the thing, or what is the species, or like however many species that produce the toxins that are prone to making this sac buildup necessary. It's necessary, right? You have to think about that. It's necessary to build it, because it probably, triggers a cell danger response. And maybe there's other gut bacteria which you can also live with in a normal way, don't trigger that. And we don't know, but I would like to find out. And you could also do this with rat studies. You can repeat that rat study that they did in the 80s, where they have the germ-free mice, germ-free mice, and they have a mutation, and then you can introduce specific bacterial strains that you think are benign, right? That don't produce endotoxin, like gram-positive. Put some gram-positive in there. You put some bifido, maybe. You can sort of build up. Of course, you can never get to a full biome that way, but even to find bacteria that are not causing it would also be a huge discovery, because we have no distinction yet, because the endotoxin stuff that I talked about was done not in an actual animal model, but in the petri dish, and they're just putting isolated endotoxin, which we could also test, right? Have the germ-free mice, just give them endotoxin purified. What happens? Give them Candida. Does that cause cysts? Like, can fungus do it too? Or is it actually endotoxin? Because the fungus in cancer doesn't produce cysts, produce tumors. So not entirely sure, like, if it also can produce cysts, probably, but we don't know. Hasn't been tested. And it's very unfortunate that this whole area of science has been abandoned, because I think this is actually the only area, except for gene therapy, that can lead to a promising outcome in PKD.
Yeah, and it's wild because, like you say, that Professor was it Seafried that did the original book, right, but didn't ask the follow-up or the correct question. And then we've got this new one, the Cancer Resolution, right, by Mark, who is asking that. Who you as you said, it went in front of a panel. And you said he's not a science guy, and the scientific panel went, this is super valid, which for anyone that doesn't understand, that's a big darn deal for a scientific panel to take someone who, graphic design he wasn't he? You know, and go, this guy's actually valid in the field of cancer research. That's no small feat. To take that so seriously. And so you're right. I think we definitely need to be spending time towards that. And it's interesting too, like, I don't know about you, but anytime I jump on Facebook, it's like, they've found the microbiome that causes MS. They found the microbiome that's caused Parkinson's disease. And so, like, they're looking at this stuff, but I haven't seen it for PKD, and I haven't seen it so much for cancer. So, and if the fact that those ones seem more specific, and that we know ketogenic diets in those cases more so than some of our autoimmune diseases really will put the reins on stuff. There's a link here that we need to look at. And to your original point, I think it's also really good to mention again that the cysts that form in polycystic kidney disease are probably the body's natural way of removing these toxins that the body, due to the endothelial damage, that it doesn't have the ability to do. I think rephrasing it like that, apart from makes us far more friendly towards our body, I think is when you take that approach to the research, you start to look at what's in there and how, you start to address this problem in a very different way when we're looking through that lens.
Yeah, and they actually, there have been efforts before. I think that was also in the 90s that the final study of that was done, but they have tried to find the cystogenic substance in cyst fluid, and they haven't been able to say what it is. They have just been able to concentrate it. So so what we know is that if you take cyst fluid out of a PKD patient and you put it on non-PKD tissue, it will still grow a cyst. So at some point, the toxin load becomes so high, I guess, that you don't even need that permeable cell wall to create this this cyst, because the body's sensing such a high toxin load that it has to make a cyst. It has to contain it.
That very talks towards, I don't know if you've gone down the rabbit hole, senescent cells, but that talks very much about that senescent cell model and SAPs. So the cytokines that get produced out of senescent cells will infect their neighbours almost like an infection and make them. They did this with Parkinson's disease rats. They just took the cells and injected it into a healthy rat, and those rats went on to get Parkinson's disease because of the cell type and the way that it manifests in the body. That I'm not saying it's the same mechanism, but it sounds similar, where we get this senescent overload where the cell, and it is that senescence is part of the cancer. So it's the same process that you're talking to, where the immune system no longer can clear the cells because we call them zombie cells, because that apoptosis dividing, they just keep dividing, delete out all their good DNA, the telomeres get so short, and then they should die and they don't. They're like these zombie things, and then they can infect with these SAPs, like almost an infection. They infect the neighbouring cells. So maybe there's a mechanism sort of similar to that happens in this endotoxic bacteria producing stuff that, spreads that way. It's so fascinating, isn't it?
Yeah. And it's always interesting to see like how many things that we thought of as sort of errors of the body finally first have a reason, and then second are tied to some kind of invader, some kind of infection. So interesting. I mean, like even the simplest stuff, even if you have like a pimple, like I do, if you get like ozonated olive oil, right? That stuff disinfects everything, and it's fat-based, so it goes into the tissue. So like all of these, like I keep noticing this, like bacterial infections, they will sort of block the body's natural detox reaction because they want to stay in there. And then as soon as you're able to kill whatever it is, then the detox reaction works, pimple goes away, or, and then also, if you're sort of, if you have food poisoning, right? So interesting. I had food poisoning like 2 years ago, last time. We sort of bought like frozen burger patties, and they had like all this snow inside the package already. And I didn't catch the problem. In Sweden, actually, I thought they were, top notch, but obviously
Swedes are trying to poison you.
Yeah. And we prepared it. Sort of got food poisoning from it, but it was so nauseous, couldn't get it out because the bacteria were blocking it. As soon as I took activated charcoal, came out like 5 minutes later.
Yeah, right.
It's interesting because I didn't realize that, because like the bacteria, they're making you nauseous, but they're also preventing you from actually spilling them out again through the tube. And as soon as you bind them up, you would think the problem goes away, you don't feel nauseous anymore, but no, it's still got to get out. But you're not paralyzed in the gut area anymore. Another thing that bacteria seem to do, they sort of paralyze the normal bodily functions.
Yeah, they're sneaky little suckers, aren't they?
Yeah. Interesting. Like so many connections there.
So many connections, and I think it's potentially more even works out into our autoimmune diseases and other bits and pieces. And I find it fascinating. I was talking to someone about this the other day, PCOS. I was just talking to my PT about it the other day. She was asking, really, ketogenic diets, we like carbs, people loading up. And I'm like, ketogenic diets, I just see them wonderful for a huge percentage of people, PCOS or any of these. And so such a great percentage of our population have become metabolic due to the Western diet, where we just can't use insulin correctly anymore. And these ketogenic diets, and just even if we just sit in a level 1, where I do the same thing, I sit in ketogenic diet. Everyone's like, how do you not eat that many carbs? I'm like, I just feel like crap when I do, like.
It's like overload.
Yeah. So fascinating, Felix. Now, is there stuff that you want to cover that we haven't covered? I love these rabbit hole conversations with you.
Yeah, we went all over the place, which is great. We could always do other topics, but yeah, I think No, I think that covers it. Yeah.
Amazing. So, what's next for you with this discovery? Because obviously, when you first discovered the ketogenic diet, you went over and wrote a book about it and then started this amazing community. Is this something you're still teasing out and working with? Where do you feel like you want to head with this next to really? Because I know your brain, you won't let this rest. So now it's just
Yeah.
like, okay, I need to figure out more. So, what does that look like for you?
Yeah, I really am trying to think if there's anything that I can find or test that would confirm that mechanism. But for now, I think we do need some studies on this, and there might be an opportunity for that that's opening up in like maybe this year. I'm like it's going to be a new like PKD patient center where patients can actually suggest things to study.
Oh, cool.
Yeah, it's like it's been originating in the Friedman lab in Seattle, and I've been in contact with Bino Friedman for since I've been invested in this whole PKD science area, so it's been a very long time. So I hope that maybe through them we'll be able to do some replication there,
Amazing.
or at least get some access to some population data where we can actually follow up with people and see if they have PKD on ultrasound or not. So yeah, so those are the two avenues I'm going down. I'm trying to get the brother tested of my client, find out if he's the guy, and if not, always myself trying new things. I'm also doing the self experimentation, doing some fungus treatment, seeing if I see any changes. But yeah, I think for now we're sort of working in the dark regarding the whole endotoxin issue, because it's so hard to test in an n=1 setting, and I haven't come up with the way to do it yet. So for now, I'm trying to reach out and find contacts within the scientific community that might be able to pull something like a study for this off.
Yeah, amazing. And are you still working with patients one-on-one, or are you doing more of that big group stuff through your website and Facebook group? How does that look for you now?
Yeah, I mostly do one-on-one. Of course, I'm active in the group, but yeah, it's always difficult to give qualified advice if you just have a single Facebook post. So it is mostly about the one-on-one. Yeah.
Great. Well, for anyone that wants to reach out to you, the best way to do it is to go to revertingpkd.com and then schedule. I think you've got a free appointment on there that people can do and connect with you and go from there, right?
Yeah, right up top is the book button. Can find yourself a good appointment that might, even with the time zones, it's all done correctly in there. Sometimes if you have to call Australia, it can be a little difficult. Well, yeah, and then I sort of talk through their current, like where they're at with patients, and if they decide to go on the program, then it's sort of explain everything, how it works, and sort of get them on like the ketogenic protocol, but then also talking through their lifestyle issues, like the everyday things that can sort of throw you off, and then later stages, also include like supplements, lifestyle, biohacks. So all of like, we have to optimize everything in life to sort of lower that toxic burden and increase the capacity to handle that stuff.
And you mentioned this earlier, and I just want to let people know what's possible with PKD is, like the fact that we can see in an ultrasound that cysts are reversing. You're saying you're starting to see that with some of the people you've been working with, which goes against the narrative that is out there around PKD. And that's why I love what I do, because I get to talk to all these people that are reversing the narrative of kidneys don't heal and kidney disease is not curable, PKD just progresses. We now am seeing so much of that where we can challenge that narrative. So I want to thank you personally for being part of that narrative and being that whole self. I always say, what do they say? Like a master is someone that studies something for is it 1,000 hours in a certain amount of time? And I go, you can be a PKD doctor, but you're never going to get the same amount of hours as if you do it yourself and live and breathe it. And that tenacity that you have to heal and then want to help others to do that, I think is one of the best gifts we can do when we're given a disease diagnosis. So hats off to you, Felix, for doing that and helping so many people and having such amazing results.
Yeah, well, thank you. I do think we can actually be a very good accomplice to any kind of doctor who's working with PKD patients, and offer a lot of insights on how to optimize life for this, and start, stopping cyst growth, reversing cyst growth, increasing kidney function, keeping it stable. Anywhere in that area is all a win.
Yeah, I totally agree. All right. Well, thank you so much for your time. I definitely, when you start to follow up on this and get some more information, you are going to send me another email and we're going to do a follow-up to this. And anything else that you want to do, yeah, please do. Anything else, I'm going to connect you with someone that might be able to do some of the scientific stuff for you. And again, anyone listening, if you want to reach out to Felix, he's great. He's got the Facebook. I'll put in his Instagram, his Facebook link, and then his main website, which is reversing. So the word reversing, pkd.com. Remember to hit subscribe and like. That way you will get notified anytime we put up more content. If you have questions for Felix, put them in the comments below and I can pass them on. If you want to know more about what we do, head to www.kidneycoach.com. Thank you for being part of our community, Felix. As always, thank you for your knowledge and your passion. And I look forward to talking to you next time with an update.
Yeah, thanks for having me. It was a great time.
Yeah, bye.
Bye-bye.